Okay, this is the last of the lessons. So it covers mutations and sequencing. I think that's pretty well it. So I'll try not to get all teary-eyed. Let's go. We've talked about mutations before. This is going to talk a little bit more about some other types of mutations. So mutation, like we saw before, is a permanent change in the genetic material of an organism. It's positive in that it can allow for genetic variation. Ooh, sorry, I'll get the pen out. So sometimes mutations can be a positive thing, which can allow for natural selection. Sometimes they may lead to negative things like diseases or disorders. Some may be harmless. There are spontaneous mutations and there are some that are induced. So spontaneous simply means that it arises as a result of molecular interactions in the cell. It just kind of seems to happen. Nothing really is causing it to happen. So it happens spontaneously, usually during one of the three processes that we talked about. So either DNA replication, during the transcription process where it's being converted into a message form, messenger RNA, or in translation, that there's errors in the amino acids that are put together. Induced kinds are those that are caused by some kind of an agent or event outside the cell. It's called mutagens. Mutagens may be physical, like UV, or they may be chemical, like carcinogens. Okay, so we have things like viruses or bacteria or things like that that can cause mutations to arise as well. So we don't have to know a whole lot about them. Okay. Chromosomal mutations are ones that there's a mistake that may involve more than one gene. So we talked about these in the genetics chapter, where it could be an inversion, translocation, duplication, or deletion of whole sections. So chromosomal mutation involves more than just maybe a certain nucleotide. It is like a gene or more than one gene. A gene mutation is a mistake in that single gene, so an incorrect sequence or message. So it could be a deletion, an insertion, or a substitution of a base pair. So chromosomal involves more than one gene, like a section of a chromosome, whereas a gene mutation is within a gene itself. Okay. If it's, we're going to look at pictures of these too, but if it's a deletion or an insertion, then it causes a shift. It's called a frame shift mutation, where it shifts the codons. Okay, so if you look at the example that's given, this is what makes sense. Okay, this is the normal codons. The fat cat ate the big bee. But if there's a frame shift, then it's going to shift the codons either one to the left or one to the right, and then it doesn't make sense. A germ mutation is one that happens in the sperm or egg, the reproductive cells. that means if it happens in those, then every cell of the individual is going to have that mutation. If it's a somatic mutation, it occurs in the rest of the cells, and it's probably just going to be localized to that region. So if, say, it's caused by UV, and the UV was focused on the section on the back of your shoulder, then it's not going to affect all the other cells in the body. It's just going to affect those ones in that region on the shoulder. Those would be the kind that end up causing like cancer, melanomas, things like that. Okay. Those are kind of terms and definitions. So let's have a look. These are the ones that you learned about in the genetics chapter. So it involves, these are chromosomal. It's a section of a chromosome or involves more than one gene. Okay, so these ones you will have to know for the exam. Probably not quite as involved for this chapter, but it's to understand the difference between them. Okay, so inversion, where you have a section that kind of has come out and reinserted itself the opposite way. Translocation, where you have a section of one chromosome being translocated to a section on another chromosome, not a homologous one. Or it could be a homologous one, but normally it's to another one, another chromosome. Deletion mutation, where a section of the chromosome is lost. Duplication, where a section of the chromosome is repeated. Okay. Gene mutations, on the other hand, if you look, this one would be an example of a silent mutation. So here's where the mutation happens. ACU becomes ACC. But if you look on your chart for codons, both of those codons code for the same amino acid. So since they code for the same amino acid, it does not affect the protein at all. It is a silent mutation. Okay, so silent one wasn't listed on there, but it is another kind of gene mutation. It just doesn't really have any effect on the protein or its job. Okay, so it is a type of substitution that is considered silent because it doesn't affect the protein, doesn't affect the amino acid or the protein. Here's another substitution. Okay, so normally it's GAA, but this substitution becomes GUA. So the rest of the protein would have the right amino acid sequence, but this one changes from glutamate to valine or valine. Okay, so depending on where that sits in the protein, it may alter its function a little bit, it may not alter it much, or it may make it so it's not effective at all. So that one is a regular substitution, not a silent one. Okay. Let's see. This one, if you look, it's normally CAUUUG. Here, there is an extra G put in right here. Okay, so you'd have G, U, U, G, A, C, U, 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 G, A, C is here. Okay, so because of the insertion, because it's an insertion, it changes all of these amino acids. It causes a frame shift and changes them. Okay, if we look at the last one, the A here is lost. So because the A is lost, it's a frame shift mutation again. This one is a deletion. So from where that happens, all the amino acids have changed, and it may even truncate the protein or might make it so it doesn't get produced at all. So some mutations can be very bad. Some may not cause a lot of change to the protein or its jobs. Okay, the last little section is on DNA profiling, and this is interesting. So I usually include a little section on this, and usually have a question, certainly in the little mini lab, but possibly on the exam where you have to actually do some sequencing. So normally when you're doing a profile, let's say you're trying to figure out who a killer is in a murder investigation, and they find some evidence. The first thing is they have to get some DNA evidence. So it might be from the hair follicles in a brush, or from lip cells from their lips on a cup, or some other source of DNA. So because they only have a little bit, in order to do the studies and repeat the reactions that they have to run, they need to take that DNA and make copies of it. So, oops, I have the pen. I didn't mean that. So they do what's called PCR. That stands for polymerase chain reaction. And you don't necessarily have to know what those letters stand for, but basically what they do is they take that little bit of DNA and they put it into a machine with certain solutions, and that goes through a series of reactions in order to make a whole bunch of copies, kind of like a photocopier. Okay, so it makes a whole bunch of copies of the DNA so that they can send it for analysis. The last step is analysis. And usually that takes one of two forms. One is sequencing, so they actually sequence all of the DNA. And the second is by what's called restriction analysis. And that lets them kind of treat the DNA with certain enzymes that cut it in a distinct pattern. And that distinct pattern would be different. They'd be closer in people that are really closely related, or exactly the same in the same person. Okay, so we're going to talk about the two methods of DNA sequencing. This is the first one, the original Sanger method. This is actually what I used to do when I was doing my master's. So it was still used, let's see, when did I finish? 1999, they were probably just getting started, or the newer method was just starting to take hold, and some places could afford to have the machines to do it, but it was fairly new at the time. So this was the original method for DNA sequencing that was used. Basically what they would do is they would take the DNA and they would use a primer, or a little sequence that would kind of get replication started. And they cause DNA replication to happen in 4 different tubes. So within each of the 4 tubes, they would put a special molecule along with all 4 regular nucleotides. They would put special nucleotides that kind of stopped replication at different places. So you'd end up with all different possible lengths of the DNA that ended with each possible letter. You're not going to have to be able to explain exactly how it's done. This is just kind of giving you an idea. So because it cuts the DNA in all the different possible lengths that end with, say, an A, when they run that tube of stuff in a special gel, then the little sections are going to be able to move along the gel depending on how long they are. Okay, so the gel runs this way, going down. So the shortest pieces, or the ones at the first, end up going all the way to the bottom because they're really fast when they run through the gel. They're charged and they move towards the other charge. And so the 5' end, or the start of the DNA section that's being sequenced, is at the bottom, and the gel is read from the bottom to the top. Okay, so if you are reading from the bottom to the top, then you do so by going from the shortest. Okay, so the A is the shortest, it's going to be the first. Why am I using that? Silly, I'll just use the pen. Okay, so A, and then you move over, and the next shortest one is here. Okay, so the next letter is a C, and then the next one is over here, which is under in the T reaction, so it ended in a T. And the next one would be a G, and then a C, and then a C, and then an A, and a G. Okay, you get the idea. So you read it from the bottom to the top. That's a G. Oop. And then C, C, T. Okay. If I were to ask you to write it from 5', which is the first of it, to the 3' end, then you'd have to tell me from the bottom to the top. That's a 5. Looks like a 3, but it's supposed to be a 5. Okay, so it starts with A, then C, T, G, C, C, A, oop, G, T, G, G, C, C, T. And that's it. Okay, so you may have to do that on the exam, but it is definitely going to be on that little mini lab. So you read it from the bottom to the top, and that's the 5' end to the 3'. Now, some things about this particular sequencing method, where it required that they use 4 reaction tubes as opposed to what you'll see in the next one. We had to use radioactive labeling, so we had to deal with radioactive substances, which is, you know, if you can avoid that, that is healthier for you. But we did take special precautions, and we had to wear special equipment, and in some ways it was kind of scary working with it, but as long as you are using all the safety precautions, then you should be okay. Still, if there's a method that you can use that does not require radioactive isotopes, then that's better. The third thing was that humans had to read the sequence. Okay, so because humans are involved in the actual reading of it, there's a chance, you know, maybe you made a mistake in some of those lines. But the second method is automated sequencing. So you basically prepare one reaction tube. You still have molecules that stop the reaction at every possible length, but this time the primers themselves are labeled with fluorescent labeling, and fluorescence. We don't, you don't have to worry health-wise with fluorescence. Okay, so they're fluorescent labeled instead of radioactive. Computer is going to detect it. Okay, so the computer detects it, and then you get, this should make it bigger. You get a printout that shows all of the sequence at once. Okay, so the computer does the work for you. Let me see. I need this one. I think that. There we go. And this is what it looks like. Okay, so if you were to read it, oops. If you were to read it, it shows you the sequence right along there. Okay, from all the different peaks. So the computer does the work for automated sequencing. You just have to prepare the original tube. One reaction tube. And I think, oops. Yep, that's it. That is the end. So that's the last lesson for grade 12 biology. You have a little mini lab to do, and then you'll be doing some review activities, playing some of the review games over again if you want to, or working on or getting a chance to look at the multiple choice booklets again. You won't have time to write things down from them, but it'll give you a chance to look at the answers. Give you some time to start your studying, make study tools, and yeah, just try and make sure that you're all ready. Don't forget that you can text me or send an email to my phone. Miss Power has the address and the phone number so that you can do that. Oh, I could just put them on here. Okay, so my phone, if you're going to text me, is this number. And if you're going to email it from your email, then it's that at, have to double check with Miss Power to make sure I'm doing this right. kudomobile.com. I think that's what it was. Okay, so good luck when you're studying. Take your time. Put stars by things if you do have questions. Try to help each other, and don't be scared to text me or email me, and I'll do my best to get back to you. It's been great having you guys in class. I always brag to everyone how I'm the luckiest teacher because I always have the greatest groups. So thank you so much, and I hope I'll be in before everything's done so that I can introduce you to Charlie, but also to see you again before we're officially all through of biology. Anyway, take care. Make sure you get some rest and eat, and change your scenery once in a while when you're studying for exams. And I will stop because I know I'm ranting and ranting, but it's the mother in me. Good luck. I'll see you soon.